hand it over to Santiago to introduce Peter.
Okay, well, thank you all for coming. Well, it's really my great pleasure to introduce Peter for his QE exam. This is sort of the tradition after finishing the exam, students present their work. So Peter came to us from Case Western as a person who's extremely interested in insect behavior, neuropathology, genetic basis of behaviors, and so on. And this is what we do in the lab. Very quickly, Peter emerged as a very sort of independent and curious student. Some students come to the lab and work on the systems that we've been working on, and others, like Peter, decide to kind of forge their own path. And that has been extremely rewarding experience. And one feature that defines Peter is that he's really unafraid to take risks and explore the unknown. And he will go anywhere he needs to go to answer a question, including, the atmosphere. And so, I won't spoil the stories, but Peter has really put together a very comprehensive, ambitious, but very fascinating pieces that the whole process was really interesting because it started with Peter finding these dragonflies that migrate. They're introduced here in California. They migrate through California and they come and go here in babies. And that was kind of the entry point to this idea of looking at insect movement, which is kind of a new topic for our lab. So it's been a really rewarding experience for us to learn from Peter. I won't say anymore. Thank you, Peter.
Awesome. Thank you, Santiago. Alrighty, everyone. Welcome to my talk, which is loosely titled Uncovering Aerial Insect Movement Patterns Above California. And before we begin, I want to do a real quick land acknowledgement. So I want to thank nature and her indigenous stewards for really providing such an amazing environment to be inspired by and question. And I also really want to highlight that this work was done on land and is facilitated by infrastructure on land that was taken through centuries of genocide. So I think we really need to acknowledge that infrastructure and the human life that has happened in order for this stuff to occur. And especially a lot of this work has been done on lands of the Patwin, Miwok, Cayuse, Umatilla, Walla Walla, Washoe, Chumash, Tongva, Otojemin, Payamakawicham lands. And then also I'm using a radar network across the entire United States where people from every corner of it have been displaced. So I just want to real quickly acknowledge that. But one I'm really excited to talk about is migratory behavior. So in any real sphere of biology, whether we're talking land, sea, or air, we're seeing migration. And a lot of these animals perform some of the craziest feats that we see in nature, like the Arctic tern flying from Antarctica to the Arctic and back each year, or large mammals crossing the Serengeti following the Green Wave. It is really a widespread and important behavior and pretty mind-boggling to me that so many animals do it to such a large scale. So while we're talking about migratory behavior for this talk, we're actually going to be defining migratory behavior as a behavioral mode and not an emergent process. So we're using the definition that it is a straight and persistent movement that is self-powered, facilitated by movement or a vehicle, and that has initial inhibition of station-keeping responses. So that's a little wordy, but all it's saying is migration is when an animal goes in a straight line or gets on a current or something and maintains a heading or position in that current or walking in order to keep going. And then the critical part is if it's exposed to a similar environment with useful resources, initially it does not stop. So it keeps going despite being in suitable habitat. So that's all it means outside the jargon, but I want to make that clear for the rest of the talk. So of course, there's different migration things and we have all different ideas of what migration looks like. So migration behavior can result in very different migratory routes and patterns. So for example, we have the classic bird, overwinters in Mexico, flies north during the winter or spring, hangs out, lays eggs, makes babies, flies south and heads back to its area that overwintered again in the fall. This is called a round trip migration. So you're doing a migratory behavior, you're moving out of the accessible habitat you were already in your overwintering grounds, you're going to a new place, and you're not stopping unless you really need to land and refuel, but you're not like recolonizing or anything. And normally birds are on the wind, right? They're following specific currents and trying to go as fast and as far as possible. Insects do very similar things. So hypothetically, we have populations of moths that are overwintering on the Gulf Coast. And during the spring, you might see a smaller movement, but these butterflies are still doing a straightened, persistent movement. So even though it's a bit shorter, they're still avoiding host plants nearby and continuing further up north. And then they'll die and have a new generation. So despite it only being a one-way movement, the migratory behavior we see is still consistent. So we're calling it migration. And this pattern completes or continues, you'll get another generation. And then at the end of the year, you'll see a final generation return back. So although you do get an emergent round trip behavior, you can think of it as a series of one-way migrations with different population. It's kind of an emergent population dynamic, but not like the definition of migration, to be clear. So I want to real quickly also talk migration and dispersal are the same in this framework if the behavior is the same. So if you're dispersing via directed movement, that's straight and persistent, it's migration. If you're ranging around looking for new suitable host patches and everything, that's also a form of dispersal, but the framework, we would call that ranging movement. So many aerial insects cannot overwinter in cold environments. So we do have to see this north-south movement across many generations. And a really great example of this is the East Asia flyway, where large amounts of insects hang out in the Indochina Peninsula over winter, and then progressively expand north into China and even sometimes into Korea. And then as the temperature cools down, they move south, and none of them can really survive in the Chinese winter because it's too cold for them. So what happens if migrant populations cannot return south? That's a very interesting question. And if you're not cold tolerant and you're in a cold environment, you're probably going to die. So climate change has altered northern wing timings in the East Asia flyway. And this leads us to have a very interesting case study where we have our East Asia flyway. We see them moving up from Indochina up into China. And these are a variety of different monitoring stations we have for corn rice roller moths that they've been monitoring for decades now. And if we look in 2000 to 2011, there's significantly more moths apparent at all of these collection points. This is structured latitudinally, so higher up is north and further down is south. And you can clearly see that the previous decade, in 2000 to 2011, there's way more than in the most recent decade. And of course, there's anthropogenic factors and, pesticide use and everything. But another really critical thing that has happened is the timing of the southern fall winds has changed. So now there's actually a fairly large phenological mismatch between when there are adult rice roller moths and when the winds allow to facilitate them to take back. So we can see in these data that a large number of moths aren't returning to their overwintering sites and are really not completing this southward migration. So this is leading to overall a large reduction in moth numbers and is a great example of how rapid climate change could actually have a very large impact on migratory insects as a whole, because if you mismatch with your wind, you might actually get caught out in the cold. So this connects to an idea called the Pied Piper effect, which was originally proposed to explain across Europe and North America, these mass emergences of insects in the fall and summer, or spring and summer. And you see a ton of moths show up that aren't cold tolerant. And then you don't see any return south. So originally people thought that was happening is this was a huge migratory trap, and then they were all dying there. But this was largely disproven in at least the Eastern United States and Europe, because radar observations showed that there actually was a return migration. It was just happening really quickly on the side of cold fronts. And also, if there is such a ecological trap, selection should remove migration from the population, because if you all freeze to death, why would you keep migrating? But climate change is likely going to make the Pied Piper effect more common, because as winds rapidly change, you're going to start to see more wind mismatches. So understanding the insect migratory routes right now is really important, so we can actually understand the baseline and start to figure out how, as climate change becomes a larger magnitude and more variability in weather occurs, we can understand what potentially might happen to these insect movement patterns. So taking a step back, what exactly are aerial insects? And I'm going to define for this talk anything that is flying above the flight boundary layer or surface layer. So you can see this red line right here. And this point is at which the movement's self-powered ability is eclipsed by wind. So below it, an insect can fly in essentially any direction they want, depending on the wind heading. And above it, the insect, whether they like it or not, are going down. The wind becomes the main vector of these movements. Typically, it's very species specific and environment specific. Trees, you have a higher flight boundary layer, but it's normally about 10 to 20 meters above the ground, and everything above that is usually considered an aerial insect. These winds are really great because they allow for super rapid movement. Like going back to an aphid example, an aphid can normally fly about half a meter a second, but when they're in these winds, they're often flying 5 to 10 meters a second. So, more than 10 times their normal flight speed, which allows them to cover rapid distances. One of my favorite examples is a lot of horned borer moths on the eastern United States would disperse over 300 kilometers in a single night during the spring, allowing for very fast movement. And then also a lot of our data show even small insects like aphids are completely capable of getting out of the wind when they have the right cue. And they also do need to initiate takeoff in order to escape the flight boundary layer and get into the atmospheric structure. So this isn't just purely us looking at a bunch of insects getting blown off the side of a mountain and misreading that as a behavior. It does seem that these insects are intentionally getting into these winds and then maintaining their position in the wind at proper thermal temperatures, showing that this is an active form of movement and not just passive. So aerial insects are very diverse, but there's a few main trends we see across Europe, the US, Africa, and that is the majority of insects up there are considered micro insects. So these insects are going to be less than 0.1 milligrams. So anything smaller than an aphid. So you're thinking your aphids, your fruit flies, other kind of things that are really tiny. And a lot of times they're hemipterans like aphids. There's also a lot of dipteran like flies up there and assorted species. But interestingly, most of the work has been done on larger insects. So these are usually moths because they're agriculturally relevant. So we have very little data on actually the micro insects. And this is A, because moths are interesting and very ecologically significant. B, they preserve a lot better when you collect them in the sky because they don't instantly blow up when you collect them with an airplane and you can use a sticky trap. And then also a lot of previous work on this has been done using these dedicated entomological radars that are great because you can actually track individual insects using radar, but how they're designed with the physics, they can't actually detect anything below the micro insect, or that's a micro insect. They have to be a bit bigger to detect. So a lot of times we don't really look at them. So that is a huge consideration. And one of the reasons why I'm much more interested in micro insects is there's a very large knowledge gap here. And there's an even bigger knowledge gap when we consider how many insects are actually in the lower atmosphere. So a recent paper estimated that 100 quadrillion insects were aloft in the sky over the continental US each summer day. So 100 quadrillion is a very, very big number. And even if you, factor in, hey, Peter, that's across an entire continent. Of course, there's going to be a lot of insects. Usually we're looking at an average of 5 to 10 insects per square meter of air above the United States each day. So if those were stars in the sky, you couldn't actually see any black, essentially. They're also incredibly important because, as I've mentioned, a lot of aerial insects have cool things they do and potentially damaging things. So we see a lot of pollinators up there. We see a lot of natural enemies up there. And then we also see a ton of agricultural pests and disease vectors. Actually, a few months ago, we found the first case of mosquitoes flying malaria in the lower atmosphere. So there's a lot of very important implications for them moving into new environments and tracking these insects because they're important for ecology with huge nutrient pulses, agriculture with them being disease or pests, and then public health with disease vectors. And then changing wind patterns has some cool implications. Not only could we imagine, our previous example with a phenological mismatch leading to the destruction of a flyway where insects can't move south, but also if wind patterns change, you could imagine new habitats being exposed to agricultural pests, new communities being exposed to mosquitoes if, warmer winds facilitate a change in direction and timing. And then also, for ecologists, it's really interesting that despite all the news of the insect apocalypse and everything, really no one's been able to find a severe decline in insect abundance in the lower atmosphere. So this kind of raises the question, why aren't we seeing this? Why does it seem like the stuff up here isn't as impacted by a lot of anthropogenic stressors than a lot of the more charismatic insect species we see down on the grid? So finally, the West Coast is an important migratory route. We see a lot of insects using it, like the Western monarch, and we see a lot of birds having a really important migratory route through here. And it's also a huge agricultural hub for the United States. But there's been no aerial insect research done on the West Coast of North America. So despite everything I've been saying, California and the West Coast is really a huge black box. So I'm very excited to kind of try to fill that niche and start figuring out what's above our heads right now in California. So the goal of this proposal will be to investigate the direction, magnitude, and identity of aerial insect movements across the West Coast. In order to do this, I have three main kind of goals, and I'm trying to complement each other. So the first one is I'm going to be observing mass movement patterns of insects using the NextRad system, which is continental, which allows us to kind of see the broad dynamics of what's going on, but lacks taxonomic resolution. Next, we're going to be describing the community of insects we find directly above us using direct sampling techniques. And a small net and a balloon can only go so far. So yes, we'll get that taxonomic detail, but we lose the scope and scale of the monitoring. And then finally, we're going to zoom into a well-known aerial migrant, the common dronefly, which has evidence of migration on the West Coast, as kind of a proxy for a larger insect, which might be doing directed movements to see potential movement corridors we're going to see along the West Coast. So zooming into the first one, looking at bulk insect movement patterns with radar, we're going to use the NextRad network, which I've been talking about, which is, if you've ever seen weather radar on precipitation maps in any news, we're using the NextRad data to get this. And I'm actually using the exact same data for these analyses. I'm just filtering it slightly differently. It's really powerful because there's 143 stations across the West Coast, and they've been recording every 10 minutes for the past 30 years. So this is one of the largest data sets of animal movement in the world, which is a real exciting gold mine to be able to look at. It can detect micro insects, unlike entomological radars, because it's designed to detect precipitation. And even, cloud precipitation is way smaller than even most of the smallest insects. So we're pretty good that we're going to be detecting most insects up there. However, even though it's really good at having a huge spatial coverage and a huge temporal coverage, it suffers from taxonomic resolution. So that's our big trade-off, is we're good at filtering for insect, but which insect requires much, much more complex AI to disentangle, and it also needs direct collection back to validate it. And given we haven't collected any yet, we're just going to be looking at bulk insect movement patterns to kind of see what the entire community is doing during this chapter. So we're going to go into a quick radar crash course. And broadly, all a radar is doing is listening. You're sending out a signal and looking for its echo. So how NextRad does that is it's a scanning radar. So it chooses a fixed scan point, like 1 degree off the ground, and scans in a whole 360 arc, and then it moves up to the next angle, scans, and so on. And what it is looking for is backscattered electromagnetic radiation. I'm going to be calling this reflectivity, or ETA, for this. And all it is essentially the amount of surface area in centimeters squared per kilometer cubed in the air. So how much stuff and what is the surface area of stuff sending a signal back to us per unit volume of airspace. And this is really the bulk of what radar is doing, because it allows you to see how much of anything is given in the air. However, modern NextRad has a few other cool pieces of data we can get from it. First is it sends 2 beams of electromagnetic radiation at the same time. So you have a vertical and a horizontal. And then by comparing the difference in signal between those two, you can get the general shape of an object, which is incredibly useful for identifying insects and also filtering out weather, because weather is usually perfectly spherical. And if you've ever seen a dragonfly, you know it is not a sphere. And then finally, we can also use the Doppler effect of the electromagnetic radiation in order to get the velocity. So we're able to see if it's moving toward, to, or from the radar. And then when you have the entire 360-degree scan, you're able to essentially figure out the broad movement pattern of what's happening above us. So NextRad, although it's limited in taxonomic resolution, we get a lot of really good data that we can use to understand insects, where they're going, and how many of them there are. Now, birds are also not spheres, so we need to be really mindful of how do we detect a bird versus an insect. And there's a couple of ways we're going to be doing that. The first way is, as I mentioned, aphids fly very slowly. Most insects are quite slow. So they're going to be predominantly the main speed of the wind. There's not much variation away from wind speed and insect airspeed. However, birds are much faster and have a higher self-propel rate. So we can actually use the standard deviation away from wind to better understand whether or not we're looking at insects versus birds, which is a great filtering technique. Additionally, insects tend to be a different shape than birds. So we're able to take advantage of their more oblong body shapes to filter it and get insects out of birds from that way. Finally, birds also usually migrate at night. So by sampling during the day, we have a much lower contamination of birds than if we were to sample at night. So using all these different methods, we have a pretty good way and a fairly high confidence that we're looking at insects opposed to weather, birds, or any other object in the sky. So kind of the two main hypotheses I want to look for are aerial insects are using seasonally beneficial timings and headings, and aerial insect abundance will be structured seasonally. So those are kind of the two main things we're looking at. And overall, with the data set we're using the 19 stations on the west coast of the United States. And I'm using the years 2014 through 2024. It's a subset of the data, but I'm doing that because that's when the dual polarization upgrade occurred. So even though we have less data, we have much higher confidence of what we're looking at using dual polarization. And then each day we're going to be using the scan closest to solar noon, A, to reduce bird contamination, and B, it's a good proxy for the amount of insects in the air because it is when the convection builds and is most strong. So we see the most amount of diurnal insects out there. So a quick overview of my pipeline. I download it from Amazon Web Services, process weather out using AI, and then I sum all of the radar scans into one vertical profile. And that essentially gets an average value for each height that the radar scans across it. And then I can filter birds out. And then finally, I'll do the rest of it with statistics. So focusing on my first hypothesis, aerial insects will follow seasonally beneficial headings and timings. If you're gonna freeze to death in the cold, you probably wanna head south in the fall is kind of the general overall idea of this. So in order to do that, we're gonna subset the data by season and station. Then we're gonna calculate the weighted circular means. If we don't do the weighted means, then we're just looking at wind patterns. So we wanna weight it by how many insects are in the air during that time. Then we're gonna test for the uniformity of movement using a Rayleigh test. And then if that is significant, we're going to test for significance using a Tukey test to see if there's differences across stations or seasons. So predicted results, if we expect to see, kind of this classic idea of migration that a lot of birds exhibit and larger macro insects often exhibit in other places other than the West Coast, we'd expect a lot of movement due north in the spring. We'd see probably continuation of northward movement during the summer because insects have multiple generations. And then finally, you would see southward movement in the fall. So this is the data that we actually got back. And you can see that during the spring, there's no significance. During the summer, there seems to be no directed flight movement. And then during the fall, there's also no directed flight movement. So what's going on? And there's a couple of things that we really need to consider. First is we're looking at every single day for a 10-year period. So there's going to be a lot of variation in wind anyways across that. And then also we're looking at a community-wide scale. So just like a busy intersection in the sky, you might imagine that the bankers are going to be directed commuters, but then you're also going to have a lot of tourists or other commuters that are kind of changing different directions. So there may be directed movement, but it's just drowned out by larger noise, or it's just so chaotic, they actually don't care. We can kind of think about it as busy city street. But if we're thinking about micro insects, and we know that micro insects from most surveys across the entire world are the main group of insects we see in the lower atmosphere, we consistently see that despite clades, they don't really have a directional selection, despite having poor cold tolerance, which is kind of an interesting thing to me. So these data of no directionality is actually quite consistent with inferred aphid migratory patterns based off of collection data in the UK, and also observed plant hopper radar patterns in China. So this is not that surprising that we're not seeing any directionality. And it seems that probably the benefits of long distance dispersal or migration outweigh the winter dieback. So, one generation of insects moving very large distances into new habitats, they are probably having so many offspring in those new uncontested habitats without as many parasitoids and stuff. That doesn't matter that some of them die due to flying in the wrong direction. You have so many of them that eventually the wind pattern is going to be right to move them to where they need to keep going and so forth. So we might actually see this be maintained just because the benefit of it outweighs the risk despite not having any wind direction selection. So then my second one is aerial insect abundance will be structured seasonally. So if we think about kind of a classic migratory pattern, we would expect if, we have the population moving north in the spring or returning in the fall, which might be a little iffy, but we could still imagine that. You would expect a peak in the spring, a peak in the fall in more southern areas. When you move more north, it would probably be much tighter because it takes time for the population to move north, have a generation, and then move south. So we kind of see this bimodal pattern with kind of two further peaks and then closer peaks once you get into higher latitudes. So that's kind of the mass synchronous round trip strategy if that's the migratory route that we're observing. And you see that similarly in like the East Asia flyway. You could also see alternative migratory patterns. So you might just see a single peak. So this could either represent a really fast generation landing and emerging all within a couple of weeks each other. So you never really see a split. A lot of insects all moving at the exact same time, or it might just be a migration stage. So insects only fly through this area when the winds are right and there's actually no suitable habitat below. So you don't see a constant amount of insects, flying. Or you might just kind of see continuous insects in the air that probably scale throughout the year because as each generation occurs, you're going to have exponentially more insects. So what do we see? So in order to break down this plot, each one of these is a radar station and they're structured latitudinally. So this is our most northern station, which is out near Bellingham, Washington. And this is our most southern station at Yuma, Arizona. It is broken into the average reflectivity of that calendar or of that week across all 10 years. And what we can see is you're actually kind of starting to see a few very interesting patterns. So when you compare it to the average temperature of that week, you start to see some interesting nuances. Firstly, we can see that the aerial reflectivity in hotter areas and more southern areas actually kind of dips during the summer, which is similar to that bimodal pattern we were talking about. However, it does correspond to the highest temperatures of the year. So it's unclear whether, maybe insects are hitting their thermal maxima and they can't fly because of that, or if there is actually some form of, mass population movement out of those areas during the times when heat is most intense. However, when you get more northern, you see that you kind of see actually a pretty interesting correlation where the peak temperature seems to be similar to the peak reflectivity. So for people who are having a little bit of a hard time viewing these plots, here's another way of visualizing them per radar. So you can kind of see these patterns. Granted, this is a log scale. So even though these lines look really flat, these are still changes of 10 times the number of insects we're talking about for each of these little bumps in the air. And then it's colored by temperature. Just to kind of have a more illustrative point, we're going to be looking at these stations, which are the more eastern stations, more in the Great Basin type environments. So we can see each one of them that I'm highlighting and the colors correspond to it. And we can see that most of the stations in the south do have this kind of interesting bimodal pattern. And as I said, it doesn't look that intense, but we're using the log version of it. So this is still a 100-fold change of aerial insect reflectivity. It's quite noisy, but there's something there. And then once you get north, you see that interestingly in Nevada, there's this really beautiful curve, showing that there's more insects in the summer and it goes down very significantly during the winter. Granted, none of these places are going to hit zero because I put a filter at 50 degrees Celsius on these data, because once you get below 50, there's a higher chance of snow, and snow can actually start recording as insects. So like I was playing with Alaskan data and you see this crazy high speed peak in November and you're like, what's going on? It's a blizzard. So that's the reason why it doesn't completely disappear for any of these stations. And then interestingly, you also see some of these stations that are just relatively flat, but pretty abundant regardless of season temperature. So I'm currently making general additive models to test the significance between temperature, season, latitude, and their interactions. I chose general additive models because seasonal dynamics are very windy, curvy. They might not be linear. And we can also expect potentially the same thing for CT max, where you often see kind of a weird pattern with a tight drop off. So I wanted to use something that could account for that wiggliness of the curves. And then given the magnitude of these data, GAMs take a quite long time to run and are a little finicky. So I'm working on them. I have some preliminary data if you want to talk to me about it afterwards with the statistical tests. But for this talk, I just wanted to kind of show you the data eyeballometrically. So kind of summing up the radar, there is no evidence of main seasonally directed movements on the West Coast, which raises super interesting questions about, do we see, is it getting drowned out by local species, or is there a cool idea that there's just kind of a bumble north, that it's a random walk that's always occurring, and eventually populations move up and then south, and it's just kind of random. And then finally, insect biomass patterns do seem to be strongly influenced by season, site, and temperature, but I can't give you any p-values quite yet. So moving on to my second chapter, describing the community of aerial insects above California. Just kind of refreshing you guys about aerial insects once again. Majority of them are quite small. They're often understudied and damaged during the collection. A lot of previous studies use sticky traps. And if you've ever tried getting anything off a sticky trap, you could imagine getting a midge that is 2 millimeters long off of it is nearly impossible. So they're often systematically misunderstood and not heavily intensively sampled because of that. And then most insects are agricultural pests. So this is really important because there's a lot of real world application to these data that we're collecting to understand who can move. But it also biases a lot of our data, not looking at biodiversity, but focusing on the agricultural pests. So we're probably actually not encompassing the true diversity of the aerial ecosystems in a lot of these studies. So critically, no aerial sampling has occurred on the West Coast, making this a really interesting question. And how are we going to directly sample these techniques? So direct sample is needed to validate observations and identify species. So we need to confirm that in the air, if we see radar signatures of insects, that those are actually species. And we need to also see who's up there. In order to do so, we're going to be using nets that are very, very fine. So the nets are a much better option than sticky traps because it doesn't damage micro insects the same way. We're going to be essentially sky trawling. So we're going to be just like the marine biologists have their net down, in the water column, picking up the plankton. We're doing that, but inverted. And then I really chose these methods to prioritize micro insects, even if they're less affected or, if they're a little bit more finicky and have a smaller wind range that we can fly. So there's two main ways we're going to be sampling. The first one is using something called a helikite, which is essentially a fancy balloon with a wing on it. So it makes it more stable during flight. And that actually came in today. So we're going to be very excited about that. And then given that the helikite is a tethered balloon, it's only legally allowed to fly 150 meters above the ground. But we frequently see that insects are over 4,000 meters in the lower atmosphere above California. So in order to mitigate that and not be sampling just like the bottom 5% of the entire air column, or way less than 5%, we also are going to do supplementary flights with paramotor. This is an ultralight-weight aircraft, which is useful because there's you're allowed to put nets and stuff on it without the FAA being regulated. But it's also really nice because they're quite safe. They have a very slow flight speed, so you're not going to be splattering insects when you make contact with them on nets. And also you can fly to those higher altitudes, but I'll talk about them more in a little bit. Oh, sorry. So kind of here are my main sampling goals. Who is up there? What is their phenology? And also really how many insects are aloft in the Central Valley? So given the data we can collect, knowing the wind speed going through the net, we can really get a good baseline to compare to our radar. And then we can say, okay, so this amount of reflectivity at this time is how many insects we caught at that same time, because we can sample within the radar range, and then we can extrapolate that out to figure out total amount of insects and whether it's consistent across the continents. So overall, my sampling plan is we're going to be sampling this year to just figure out how the balloon works and everything at kind of 3 different sites throughout the Central Valley, mostly going to be basing out of the Jepson Prairie Preserve. And we're going to be sampling in spring, summer, and fall. I think it's really important to sample at these because one, they have very different temperatures throughout the season. They have very different plant availability. And also the Central Valley's atmosphere is quite different depending on the season. So it'll be really cool to see how that all changes to result in different insects. And we're going to occur sampling at least 3 different times per day. So we're going to be sampling midday, like our radar observations, at night, because there's a lot of evidence to show there's very different communities during different times of day. And then finally, we're going to be flying at dawn because one, there's a third little flight of insects you see during that time, but also that's the most stable, safe air. So if we're going to be using the paramotor, that's really the only time you can fly it safely. So we want to sample at the same time so we can get that direct comparison to see how insects change across the air column. So one thing that's really cool about these data is we're in range of the NextRad system. So we can actually see what the average airspace looks like throughout the seasons and everything, which can help inform our sampling plan. But during the dawn specifically, it's nice because we can get a little smorgasbord of all the insects up. So during the night, a lot of nocturnal insects will actually stay aloft during the dawn as convective thermals raise the part of the atmosphere they're in. So you can sample nocturnal insects during the day. You can also sample diurnal insects taking off. So even though you can only sample during the dawn, it gives you a little bit of a snippet and it's the safest time to fly. So going off the preliminary data, we conducted one flight on September 13th. I had a paramotor flight instructor take me up, and we sampled from 400 meters from the Davis Airport flying around Woodland, which was a really cool experience. We had a 12-inch diameter net sticking out the side. Granted, the net we're going to be using for actual surveys is significantly bigger, and this was a butterfly net I used in the field. It has quite large threads, so a lot of insects kept going through. Like one of the insects I caught was already halfway out escaping, or just hit the net and pushed through. So we likely are really undersampling the amount of insects in the lower atmosphere. But even with all these things considered, we caught 3. So we caught a female sarcophagus, 3 insects in 30 minutes. Let's go. The funny thing is even when you compare the cost of flying and the amount of gas we did, this was actually more effective return on insect collection than my field work using a car based off of gas mileage. But we caught a ceratopagonid for non-entomologists. This is a small biting midge, which is cool. We got a female tephritid, which is a small flower fly, and it's actually specializes in a native California host plant. So it's cool that we kind of got a little native action. And then we finally got a thrips. And if entomologists would like to know, or non-entomologists, thrips actually can't really fly. So, even though we're talking about, insects actively being up there, kind of weird finding an insect that can't fly very strongly 1,200 feet above the ground. Finally, the one really cool thing is you see this. This is actually a radar cross-section of the exact time we were flying during that period from this area. So this is the real density of insects that we were experiencing and flying through. And I put a little dotted gray line right here, and you can see we were flying through fairly dense insects, but there was also, this nocturnal layer above us. And if we flew a bit lower, there'd be a lot more insects. So it shows that we can actually use hindsight radar validation to really confirm where we're flying later on. So it's going to begin super soon. We got the balloon. We need to build a few more things, but we'll be sampling probably in the next couple of weeks. Also, just for other researchers, word of warning, tariffs come into effect when they drop off the package and are not cost during shipping. So yesterday we showed up and UPS won $400. So just be mindful of that. These data will be the first aerial survey of insects in the lower atmosphere. And also using these data, we're going to identify species of interest, phenological patterns, and baseline for our Western weather radar systems. And then finally, we're going to look at testing for evidence of the common dronefly. So one thing that's nice about the common dronefly is it's a macro insect. It's much bigger. We see it, and we know that macro insects tend to follow different patterns than micro insects. So looking at radar on the UK, we see that macro insects like droneflies are actually much more directional with seasonality. So you can see that during mass movements, there is downwind migrations heading south and north respectively. So it's a very interesting thing to look at. Although they're only 2.5% of the aerial insect abundance, they tend to be, natural predators or larger insects. So it's still important to kind of consider. I really like the dronefly because there's mass observations of them moving north in California multiple times. No one's ever actually seen a return flight though. Also, we've caught droneflies at high altitudes, like 2,000 meters above the ground in previous sampling studies throughout the world. So we know that the species normally does that. Finally, they also are very well documented to have super female sex bias migration. So it's nice as kind of a sanity check that if you see thousands of female dragonflies showing up at the exact same time and then disappearing pretty shortly, there's an evidence of a mass migration occurring, which is, not perfect, but still a nice little sanity check. So we're going to be using stable isotope analysis to do this. Essentially, based off of the weight of the hydrogen in water, you're going to get a slightly different pattern in the isotopic ratio of precipitation depending on where you are in the country. So this gives us not a very fine map, but a general map of origin using these flies. And last year, I drove across the entire state a lot of times to collect a bunch of water samples and flies. So we have a general baseline of these. And this will allow us to kind of identify the origin of these dragonflies. So the hypotheses for this chapter are dragonflies perform seasonal migrations through California. Female dragonflies are more likely to migrate with males to make sure that the observations in Europe are fairly consistent with the ones here. And then also I'm hoping that dragonflies follow consistent annual routes. So I'll be collecting data from multiple years to kind of see if there's a general pattern emerging. So for collection, last year, it turns out that UC Davis is by far the best place to collect dragonflies when you weight it by catch per visitation. So I spent 6 months driving from Lassen down to Irvine, and it turns out the Davis Arboretum is probably the best place to collect. We're going to do biweekly surveys from UC Davis from March through November. And we're going to do this year and next year. So we'll have a 3-year data set of isotopic values to kind of get a general pattern. And then collected flies will be sexed and used for stable isotope analysis. So a general prediction we have is if the flies are not migrants and are coming from the same area, like resident flies, you would expect their isotopic signature to match that of the groundwater nearby. Because they develop in water, it should be a pretty good ratio. And in other species closely related, we see a very similar pattern. We still need to do the calibration curve for this species. So more on that later. And then if there's migrants or large dispersal, we should expect a large mismatch. So the flies are not matching the water values of the area we collected them from. So this is preliminary data from the example flies, about 15 flies I collected from April of last year to August of last year. And what do we see? We see that there's actually a very large mismatch between the water isotope values of the flies and the groundwater, suggesting that they may not have originated there. Interestingly, Lassen Peak had a fairly tight connection between the two, which is somewhat unexpected, but I thought was pretty cool, and I'll talk about that in a little. So if we see this big mismatch, where are flies supposedly coming from? In order to understand this, this is a quick idea or crash course on stable isotopes. Is each of these red triangles is where we collected it from. And we created this map using a spatial general linear mixed model that factors in longitude, latitude, elevation, and a few other factors. And it takes those values of the area we collected at and extrapolates it to the rest of the area. We're going to be adding significantly more data points to this map and a much larger temporal range to this map too. So it's only going to get better from here. So we take this and then we take the isotope or value of the fly and we give the model and say, hey, where is this fly most likely originating? And this is an example of one of the flies we collected at UC Davis. And what this means is the p-value of 1, it's saying the fly likely originated in the green. And by the time it drops down to 0, it's saying there's a very low chance the fly originated here, is essentially all these maps are saying. So for example, this fly was collected around UC Davis, which is right around here. And what it's saying is, I don't think the fly originated here, but it might have originated in the foothills of the Sierra, like this green band, or the Great Basin, which is kind of interesting. Why would a water-loving fly be in the Great Basin or originate there? And when we look across all the flies we sampled, regardless of where we collected them in the state, even down in San Luis Obispo or up in Lassen or in UC Davis, they're all having a similar isotopic origin, which is kind of this foothills, coast rangey area or the Great Basin. So I don't really know how to interpret that yet. We need to rerun our calibrations on the flies and add more data to these isotopic maps. Potentially the irrigation might be a huge factor. That's water brought in from the desert or the groundwaters that aggregate like rice in the Central Valley might be a huge factor. So we need to sample more there. But what we're seeing right now is there's a very large mismatch between the flies. And as of now, it's saying they're more in the foothills or out east, which is a very interesting dynamic I'd love to talk to more people about. But take everything with a grain of salt because we need to do a lot more calibration and get a lot more data points. I just want to show it with you guys. And then also, I have not analyzed the fall collection yet. But if we look at the flies we collected throughout the year, there's very low numbers and there's usually an even amount of females to males. And then we get to October and it's hundreds of females we collected in October and November, and they just popped up one day and it's incredibly sex biased. So I'm very optimistic later on. We're currently analyzing the 164 females we collected this year, which might give us a better idea of potentially what this dynamic is happening. So real quickly, a summary, stable isotope data suggests that they do not originate in the sites we were collecting, but their isotope values are similar. So they might be originating in the same place or using similar resources with irrigation. Fall data still needs to be analyzed, but it's really cool to see that massive influx of females, which makes me kind of excited. Species-specific calibration curves for the stable isotope needs to be done. And also, we're going to continue collections to understand how annually consistent these patterns are. So kind of overall, the summary is we do see that aerial insects do see these kind of seasonal biomass patterns that are seasonally structured and probably influenced by temperature, but there's no bias in movement direction. Chapter 2 shows this is the first aerial survey in the west coast of North America, so we're really excited to see who originates there. And then finally, hoverflies do show evidence of migration or dispersal or ranging behavior, but we need to really get more data down. This is just preliminary data, but it's really exciting to see that there is such a strong mismatch, because I was really not expecting that to occur across all sites and all seasons, which is really unique. And then finally, I want to acknowledge my lab, specifically Santiago, for being so trusting that I could go rogue like this. And I also wanted to thank everyone in the Eve department and my fellow CPB students. I also want to thank the UC Reserve Network and the Sentinel Site Network, the National Park Service, Blue Sky Paragliding, Collins Pine, and they were great because they gave me field sites, they've given me funding, and they've given me guidance. So I really couldn't have done this work without this huge network of natural reserves and people excited about bugs in the sky. And then finally, I want to thank my entire QE committee for giving me guidance, given I'm kind of in new territory. I've really leaned on their expertise for ecology, movement, and insects and radar. So I couldn't have done it without this process. So thank you so much. And I'd love to answer a couple of questions if we have time. Yeah, Brian.
Yeah, that was really cool. So I'm curious about whether there might be a way to kind of use the information from the aerial sampling to inform the analysis on the radar data.
Yes.
And what I'm thinking about is if you, okay, you collected 3 insects, but 33% of the insects you collected are non-flying.
Yeah.
So, if that's kind of suggesting something real, which is that there's a huge chunk of insects up there that are actually going to be incapable of directed movement, I'm wondering if you can use the identity of the insects that you collect to refine your filters in the radar data to actually try to only capture the types of insects that you might think are capable of directed.
Yes, that is a really great question. I'm just pulling this up because yeah, you're right. Like across almost all sampling we've done, Thysanoptera, which are those flightless thrips I were talking about, are fairly frequently higher up there. So I think getting the baselining these radar data with aerial sampling is really best practice for a few reasons. One is you can use it to get an accurate biomass estimate. So you can actually use it to baseline how many insects you see up there. But also the other thing you can do with it is there's some really cool papers that show you can actually get different cluster the insects out quite differently based off of their radar signatures, but it's hierarchical clustering. So usually you get insect group 1, insect group 3, insect group 4, whatever. So what will be really cool is using that collection data to correlate those clusters with the insects you directly collected, which could be really informative for, as you said, like if there's one that really correlates with strong radial velocity, which is movement speed, you could definitely use that to correlate, find which cluster has the most amount of moths that can do self-powered flight or something like that. So I think that those chapters all feed really nicely into each other, and the more sampling we do, aerial sampling, the better and more nuanced insights we can get from the weather radar. And then the weather radar can tell us what are the best times to sample. So they really kind of play into each other quite well. Oshi.
Really great work, Peter. I'm really interested in like what does predation look like for like hoverflies or like aerial insects in general, and like food sources as well, because I like those also change seasonally, and I expect like change to affect those. And how does that play into any of?
No, I think that is a really interesting question, and I'm gonna kind of answer in two different ways. So when they're in the lower atmosphere itself, there's not that much evidence to show there's a lot of predation from other insects. You know, they're still only at a couple of meters a second, and they're all getting blown downwind. That being said, we do know that migratory passerines eat a ton of aerial insects in the air. We see that moth migrations are actually strongly or Mexican free-tailed bat migrations are strongly correlated with migratory moths. And we actually see really cool evidence of harriers in the Indian Ocean eating dragonflies as they cross across the Indian Ocean, which is mind-blowing. But when we see the overall community, we do see some really cool things. So the overall, most insects are usually agricultural pests. So we see like usually during the day, most of them are aphids. But like hoverflies, their larvae actually eat aphids. So we might think that hoverflies might be co-migrating with the aphids in order to keep track of those pests because their larvae, eat them. We also see a ton of ichneumon wasps in Hymenoptera, which are natural enemies. So they're parasitoids. So we actually might be seeing this very large co-migration dynamic going on where everything's following these anchoring herbivorous insects. And what's even cooler is there's a cool paper from Africa that shows that if a moth, their migratory moths land and F1 still experiences some predation, but they experience 40 times less parasitoid pressure than F2. So it seems like it's constantly running. You got to keep moving or else your parasitoids build up, your natural enemies build up in large enough numbers. So we might actually see this kind of cascading race across the landscape where the aphids are trying to escape and all their natural enemies have evolved to follow them. So I think there's a really cool dynamic in there. I want to talk a lot more about it, but we don't know who's up there. So we can't really ground any of our hypotheses yet on the West Coast. Marissa, I think you raised your hand first, right?
Yeah, so I'm wondering about your surprising results on the radar that you didn't get any prevailing movement.
Yeah.
And I'm wondering about the historical consistency of prevailing winds in this region compared to the regions where people do see these mass migrations. Is California just more stochastic in its, or the West Coast, I guess, more stochastic in its wind events compared to that Asia subcontinent to continent migration? Is that a possibility?
That is actually very interesting. And, just like all weather, wind is actually quite stochastic everywhere. So essentially anywhere you look, you can find wind vectors of any given direction. But it is very site specific. So like you see, some actually quite fixed wind patterns on the West Coast due to the topology and relation next to the ocean. I would say from what I've gathered, they have different wind patterns on the West Coast because of proximity to, the Southwest and the ocean. And also like the Central Valley has some very consistent wind patterns. But just like across everywhere, like if you look at the wind roses for any given site, there's always going to be more wind directions in certain areas versus more wind directions in other directions. So given that we saw no consistency anywhere is actually really interesting because there are prevailing wind patterns in these stations. Denise, you raised your hand?
Yeah, I don't have any questions. I actually just wanted to give you a loud congratulations because I feel like as a lab, we've seen you navigate the ups and downs of entering a whole niche of research. And I think you did a fantastic job presenting your data. So I just want to say that.
Thank you. Thank you. And in that case, we're already 10 minutes over. So thank you guys so much for the time.